Hello, everyone. My name is Lynn Shearer, and I'm a publisher at Elsevier. I would like to welcome you to this webinar hosted by the journal Current Opinion in Biotechnology, entitled Single Molecule Imaging with Multiple Wavelengths. Thank you very much to our sponsors at Andor for making this event possible. Before we begin, let me remind the audience that we will have a questions and answers session following the presentations. Please submit your questions at any time during the presentation using the Ask a Question button on the right-hand corner of your screen. I would encourage you to input questions as and when you think of them. These will be addressed in the Q&A session at the end. The more questions asked, the better the session will be. It is my pleasure to introduce and welcome today's first speaker, Dr. Stephen Webb. Dr. Webb is a senior scientist in the Central Laser Facility at Rutherford Appleton Laboratory, following a PhD combining fluorescence lifetime imaging with structured illumination microscopy at Imperial College London. He has developed single molecule and super-resolution techniques for a wide range of scientific problems, in particular studies of the epidermal growth factor receptor family of membrane proteins. His current interests include multifocal imaging and the use of adaptive optics in microscopy. Without further hesitation, I would like to hand over to Dr. Webb to begin his presentation.
Thank you. So I'm going to be talking about single molecule methods in cells. But first of all, I'd just like to explain a little about the place where I work, because it's quite an unusual scientific institution. So I work in the Rutherford Appleton Laboratory, which is in Oxfordshire in England, and we provide central facilities for a whole range of scientific methods and techniques, not just for the UK, but for the whole of Europe. So we have synchrotron facilities, we have petawatt laser facilities, we have the ISIS neutron source, and I'm housed in the research complex at Harwell, which is what is described as a research hotel, where people come for a few years with a research grant, and they make use of all the facilities on site, including our own suite of advanced microscopy facilities. So our facility is called Octopus, and we have calls for access every 6 months, which are peer-reviewed. And as I said, they're for researchers from both within the UK and from the EU, but we do also provide access on a commercial basis as well. And we have a whole range of fluorescence and other optical microscopy systems, not only the single molecule systems that I'm going to be talking about, but also total internal reflection fluorescence on its own. We've got structural illumination, STED, and single molecule localization, super resolution microscopy techniques. We've got optical tweezers, we've got multiphoton, time-correlated single-photon counting, FLIM, etc., and you can also combine a number of those techniques into the same microscopes. And while you're here, you would get access to cell culture facilities, simulations, whatever you required, essentially, that is required to make your experiment work. And you would get paid as well for travel and accommodation and so on, as I've mentioned. So it's a really unique facility, and the things I'm talking about over the next quarter of an hour are things that you can apply to use. So we have our own in-house research program, which is particularly focused on the epidermal growth factor receptor family of membrane proteins. And these are variably called EGFR or HER or ERB. You'll see all of those names, they're all the same. And there are 4 members of this family, so ERB1, ERB2, ERB3, ERB4, and they are involved in a number of different cancers. And there are a number of drugs, therefore, that target these proteins, including, for instance, Herceptin, which is a breast cancer drug. But the problem is that we don't actually have very much understanding of the fundamental mechanics, if you like, of what's happening during the cell signaling process involving these receptors. And the consequence of that is that drugs like Herceptin, we can't tell in advance whether it's actually going to work for a specific patient. And there are horrible side effects as well, because we have such poor understanding of this protein family. So there's been a lot of work in the past in trying to determine the structure of these proteins, particularly using X-ray crystallography, and there's been a lot of success using this method. But the problem is that if you're doing X-ray crystallography on a transmembrane protein, you can't look at the extracellular domain and the intracellular domain at the same time, because you can't crystallize the entire structure. And similarly, there's been work on electron microscopy as well to look at the structure of these proteins. But to actually understand what the conformation of the entire receptor is on the cell surface, and to see if there's some sort of conformational link between what's happening in the extracellular domain and what's happening on the intracellular domain, and to understand how these molecules are organized in actual cells. You know, are they in lipid rafts? Are they in some other sort of signaling platform? You need to use fluorescence microscopy, which is ideally suited to imaging live cells in a way that crystallography or electron microscopy aren't. So we specifically use total internal reflection fluorescence imaging, which involves it's ideal because we want to look at transmembrane proteins. And in total internal reflection fluorescence imaging, you only image a very thin layer, just over 100 nanometers thick typically. You only excite fluorescence in that layer, and so you get no signal from deeper within the cell, either from other fluorescent labels that you're not interested in, or from autofluorescence. So the result is that you get a much higher signal-to-noise ratio and a much lower background signal. And in fact, there's also an enhancement of the signal, even from those molecules that are in the cell membrane. So it's also compatible with single-molecule imaging, so it's absolutely ideal for our purposes. Now, why would we want to do single-molecule imaging? Essentially because cell signaling it's a localized stochastic process, and if you look at the whole ensemble of receptor proteins, then you're not getting the full picture. Essentially, if you're looking at the whole ensemble of proteins, there's a lot of averaging going on, and you won't pick out the rare behaviors of individual proteins. And if you just think, sort of, as a physicist, this is the sort of thing I would think of, if you think of the different orientations of the receptor proteins, if you look at the whole population, then all the orientations will average out, and you won't spot any polarization or orientation to the global population of molecules. But if you look at the individual molecules, you can see whether they have specific orientations in specific places in the cell. And another thing which I think is really key is that usually when we're doing ensemble fluorescence, not always, but often, we have to use much higher concentrations of the protein that we're interested in than is actually normal under physiological conditions. And by doing single molecule imaging, we can go back to more typical concentrations. So on this slide, we've got two images. On the left, we've got an image of A431 cells, which has a very high number of EGF receptors. As you can see, it's got about 2 million copies in each cell. And its ligand, EGF, we've got 50 nanomolars. So this is a high concentration, and this is an image taken in a confocal microscope. And you are exciting an enormous number of these receptor proteins. And over time, the fluorescence from the fluorescent labels on each protein bleaches, and it bleaches it in an exponential fashion, as shown in the plot at the bottom. In single molecule imaging, we want to see the individual proteins. So we have one fluorescent label on each receptor protein that we're interested in, and you can see them as individual spots in the image, as you see on the right. And if you take one specific spot, you might, we call it single molecule imaging, but this is actually a bit of a misnomer. You could have several molecules in each spot. And as the intensity, as the molecules bleach over time, you'll see a stepwise reduction in the fluorescence intensity, and you can actually use that to count how many molecules you've got in each spot, which is fantastic if, for instance, you want to look at the stoichiometry of an interaction. So you might appreciate that one of the really key things in doing single molecule imaging is the data analysis. There haven't been a lot of methods in the past of analysing single molecule images, but single molecule images look remarkably like images of stars in the sky. There's just a difference in length scale, rather dramatic one. And so we have borrowed techniques from astrophysics to process our single molecule data. It's actually a lot easier in astrophysics because stars have pretty well-defined trajectories through the sky, and the background signal is fairly well characterized, and it doesn't change over very short length scales. But in the cell, these molecules are moving around, often very rapidly, and the background signal can also be quite significant. So how do we analyze our data? Well, we basically compare two models for each part of the image, one that you have a molecule, one that you don't have a molecule, that you've just got background signal. And we assume that there is a Gaussian noise over the field of view. And then we use Bayesian inference to determine the probability that at each point in the image that you have a molecule or that you don't have a molecule, that you've just got background signal. And this pair of images is just a simulation, but it's demonstrating how you've got simulated data on the left with molecules with background, and on the right the software has correctly identified where all the molecule positions are. So one of the problems that doesn't arise with stars is that fluorescent molecules blink on and off. And also you could have a dimer in your cell, so two molecules to start with, which then split into two monomers, or the opposite way around, two monomers coming together and forming a dimer. And so the software will also calculate probabilities that one part of a track through space and time is linked to another part of a track through space and time before or after such interactions like that. So one of the things that we study a lot are the association and dissociation rates of these receptors and the ligands that join to them. And we have an OptoSplit 3 on our microscope, and we excite we have three different fluorescent labels, and we excite at 488, 561 and 640 nanometers simultaneously. So that allows us to image either, for instance, two receptors and one ligand simultaneously, or three receptors, or any other combination. We're imaging them onto a single Ixon detector. So in the top row, you've got three sub-images, the three different wavelengths, and then you're looking at the three, those pink lines in the middle are the tracks through space formed by the molecules through time as detected by the data analysis software. And so we can measure the rates that these molecules diffuse and how they associate and dissociate with each other, and look at the effects of different drugs on those rates. A second thing that's very important, and this has led to us understanding how the crystallographic model had its limitations in studies of EGFR. Crystallographic models suggested that there was always a distance between in a EGFR receptor dimer. Crystallographic studies suggested that it was always 11 nanometers between the two EGF ligands on that dimer. But 11 nanometers is too large a distance to be measured by fluorescence resonance energy transfer, FRET. So when we did single pair FRET with one, we used the EGLF ligand. Some of them had some had a donor fluorophore on them, and some had an acceptor fluorophore on them. When we did the single molecule FRET, we found that we did actually get a FRET signal out, and this demonstrated to us that there must be other patterns of association between these receptor molecules. So one possibility was that there were higher order oligomers, that the molecules weren't just in dimers, but they were perhaps in tetramers or even higher order oligomers, and that led to some of the ligands being closer together than was previously thought. And the other possibility was simply that the dimers weren't actually arranged in cells in the way that had been crystallized for X-ray crystallography. And we found it's basically a bit of both. So using other studies, mainly from molecular simulations, we determined that it was the molecules, rather than standing proud from the cell membrane, actually lay quite flat, and that the molecules are arranged, and we're still working out what is arranging them, but they are arranged in distinct patterns on the cell membrane. So we've also done single molecule FRET, not only measuring the two wavelengths of the acceptor and the donor, but also looking at the polarization. So the FRET efficiency, the efficiency of energy transfer from donor to acceptor, is not only a function of the spectral overlap between those two molecules, but also the relative orientation. And so if you measure the emission polarization and its wavelengths, you can get extra information. And what's really nice in this pair of plots is that you can see that at X, there's a marked change in the FRET efficiency due to a change in separation, because there's a change in the wavelength signal. But then there's at Y, there appears to be a change in the relative orientation of the acceptor and donor. So that's really nice, and it's difficult to do. This is a particularly nice example from one of our experiments, but that is the sort of thing that you can do at the single molecule level that you can't do at the ensemble fluorescence level. Something that we've developed over the last few years is a method of imaging distances that are a bit longer than what FRET can allow you to measure, but it's shorter than what super resolution techniques like STED microscopy enable you to measure. And a few years ago, there were a couple of papers that looked at distances between pairs of molecules on glass. And the method is that you have two molecules initially fluorescing, and then one of them bleaches, and you detect the center of the point spread function before and after bleaching, and you find there's a tiny spatial shift. Now, if you do that in proteins on glass, you can get pretty accurate results and get a good measurement of the distance between the two molecules in the sort of 10 to 50 nanometer range. But if you do it in cells where the signal-to-noise ratio is much poorer, then you don't get good results at all. So what we've done is developed a technique that's based on probabilities, and so the plot on the right is showing you our best estimate of what the separation between the two molecules in such a spot is. We use a technique called bootstrapping to do this. But essentially what we end up with is a separation and an error in that separation, which is not something which is particularly common in fluorescence microscopy, getting actual errors, so this is really valuable. So if we look now on this slide, we have an image with loads and loads of pairs of molecules in it, and we determine a 68% confidence interval for each of those pairs of molecules. And then if we look at the image as a whole whole data set, we can then get a probability density function for the whole data set, which has certain peaks which correspond to certain separations of fluorophores in our system. And there are different errors for different pairs of molecules. So we can improve the resolution if we remove all those pairs of molecules that have the larger errors. And this is what we're seeing in the bottom row. So if we just take those molecules with errors inside the grey box, then we have a smaller data set, but we have a much better resolution. So bottom right, we're now seeing much clearer peaks for what the possible separations are of our receptor molecules in our cells. And we've done a number of studies with this methodology with different drugs that disrupt actin filaments, for example, and which disrupt cholesterol. And what we're seeing is that we're able to determine what the different separations are. I'm not going to go in complete details because this paper is sort of under review at the moment, but the idea is that having determined the probability density function of the different separations available or present in the receptor population, we can then determine what the different separations are, which is the blue histogram bottom right, to quite high precision. So I'd just like to then talk a bit about what we've done to try and image more probes. I mentioned earlier how we'd image 3 probes using an OptoSplit 3. It would be even better if we could image more than 3 probes simultaneously, and if we're going to do that and spectrally separate them, that means going into the near infrared. And so this is some work we did with the 2CAM. A big advantage of imaging in the infrared is that the autofluorescent background is a lot lower, as you can see here. So at 780 nanometer excitation, it's negligible. So it'd be brilliant if we can use some of the probes that are at that sort of wavelength. And here's a picture of the microscope, in fact, with both a 2CAM on a Zeiss TURF microscope and then OptoSplit 3s on each branch of the 2CAM, and then 2 Ixon 887s on there. So in principle you could image 6 colours, it's just that we couldn't find 6 that could be 6 fluorescent probes that could be spectrally separated. But we were looking at these, we did the first single molecule imaging of near infrared probes. It is difficult because the brightness of these probes is a lot poorer than of the visible probes. But we were able to see, as you can see here, we've got some nice single molecule traces of their intensity over time of both IR dye 800 CW and IR dye 700 DX. And if you can do that in EPI and not just TURF, then that suggests that you might be able to image much deeper into cells. Remember, we're using TURF to lose the autofluorescent background from our cells, where if we can go into the near infrared, we don't even need to do that. We can image much deeper. So the other thing that I'm using the 2CAM for at the moment is in our work to study adaptive optics and single molecule imaging. So, as I said, we want to go deeper into cells, but as you go deeper, the image of a single molecule becomes increasingly aberrated, and it becomes very difficult to determine whether you've got a single molecule or not. So we want to correct, but we want to know whether correction is leading to any improvement or not. So we're also using a 2CAM to split the fluorescence from our microscope between an adaptive optic system and just a standard system. So we've got 2 Ixons on the 2 arms, and so we can study how different aberration corrections impact on our single molecule images. But this is something that's very much in progress at the moment, so I'm just sort of telling you how you can use the 2CAM in a slightly different way in single molecule methods. And this is something that's going to be very valuable both for single molecule imaging techniques and for super resolution techniques based on single molecule imaging. They are closely related, and this will enable us to vastly increase our signal to noise ratio, which means that we'll be able to image more single molecules, and we'll get a much higher localization precision for each one that we detect. And another brilliant thing about adaptive optics is that you can apply astigmatism, which enables you to get axial resolution at the same time, and you can image a complete focal stack without moving your sample, just by changing the properties of the deformable mirror inside your microscope. So that's how I've used the 2CAM in my research, and indeed Andorix on EMCCDs as well. There's a whole operations team at our facility who are able to help with these techniques. So if you are interested in using any of them in your research, please do get in touch with me, and I'll stick around for the question and answer session at the end. Thank you.
Thank you very much, Dr. Webb, for your presentation. It is my pleasure to introduce and welcome today's second speaker, Dr. Fabien Pineau. Dr. Pineau received his PhD in biochemistry and molecular biology from the University of California, Los Angeles in 2007. He was awarded both a Marie Curie and a European Molecular Biology Organization fellowship to develop new fluorescent probes and microscopy techniques for single molecule imaging of living systems in the Department of Physics and Biology at the École Normale Supérieure in Paris. Since 2011, he has been an assistant professor in the Department of Molecular and Computational Biology at the University of Southern California, where he heads the Single Molecule Biophotonics Group. His laboratory's current research includes the study of nanoscale cellular compartments and their role in modulating the diffusion and the activity of proteins involved in normal and pathological cellular signaling and responses. Without further hesitation, I would like to hand over to Dr. Pineau to begin his presentation.
So it's a real pleasure. Thank you, Lynn, for the introduction. It's a real pleasure to be here and to tell you about our research. So today what I'm going to talk about is new techniques that we've developed to try to push the boundary of detection of single molecule all the way towards the animal imaging. And as a follow-up on Stephen's very nice introduction, I just want to remind you how wonderful these single molecule techniques are. And they allow basically improve image resolutions as well as the resolving cellular nanostructures in sick cells when they are applied to super-resolution imaging such as PALM or STORM, where, as mentioned by Stephen earlier, you can actually localize individual molecule using mathematical Gaussian functions to improve the overall imaging resolution of the final image. You can also use these techniques in live cells, looking at diffusion dynamic at the nanoscale through the same principle, but this time taking into account the fact that your molecule is diffusing around, and that allows you to detect subpopulation of diffusing molecule, to classify them in different diffusion behaviors, and indeed to access asynchronous and different dynamic molecular behavior at the nanoscale. So these techniques really allow us to peer into cellular process with amazing details at the nanoscale. One limitation, however, of the current techniques and single molecule imaging are that they're really optimized for cell culture tissue slices. And there's always a concern when you work with cells that is related to the fact that your cells and the molecular events that take place on the cultured cells might not truly represent biomolecular events as they occur in vivo. And few examples or few possibilities that cell line can actually lose tissue-specific function. The fact that usually when you grow a cell on a cover slip, you have generally an absence of extracellular matrix, which might change the way the cell behave, and obviously, generally, cells in culture tend to overexpress tagged biomolecule. So there's really a need to move and to try to move our single molecule imaging from the culture dish all the way towards the animal. And so when you want to do such a move, basically it comes with obviously a few complications as well as advantages. So obviously you need to find animal models that are amenable to microscopy if you want to achieve single molecule in live tissues. And you also need to engineer different type of tissues, the aim being trying to reduce the signal to noise, try to improve your signal to noise, and try to bypass the autofluorescence that generally comes when imaging a tissue. And obviously you need to be able to express your molecules or your protein of interest at endogenous level in order for it to represent the normal behavior of the tissue. Now the advantages, so these are all complications that need to be solved. The advantages on the other hand are that you are now capable in principle to work with the native 3D environment of the cells under homeostatic control of the body of the animal, and obviously at endogenous and physiologically relevant expression level, which is key for to get a good understanding on the biomolecular process. Now, one overarching advantage of that is if you now work with a live animal, you also have access to disease model, which allow you to look at these molecular processes at different scale and at different representation or different stage of a disease, and you can think about looking at Parkinson, aging, or muscular dystrophy. And for today, what I want to show you is that you can use C. elegans nematodes as an animal model, which are highly amenable to light microscopy, notably because of their easy genetic. And a new technique for single molecule imaging called complementation activated light microscopy, or CALM, which allow a controlled and tissue-specific single molecule detection at physiological expression level by simple knock-in of fusion protein. So I'm going to start by telling you a bit about CALM, this technique called complementation activated light microscopy. This is a technique which is based on the use of a split GFP. A split GFP is basically a traditional GFP which has been split in two parts. The split GFP that we are using has its terminal 11 beta sheet fragment removed, and you end up by removing this fragment with a large fragment, the secondary large fragments of the split GFP right here, which is non-fluorescent. The absence of fluorescence comes from the fact that in the beta sheet that has been removed, there is a key amino acid, glutamic acid 222, which participates to the chromophore formation, and when removed, you end up with the big fragment being non-fluorescent. This split fusion, fluorescent protein has been developed by the group of Geoff Valdo in Los Alamos, and the very interesting thing is you can actually recomplement the protein and make the GFP active again by simply providing the missing beta sheet fragments as a synthetic peptide in solution. And so, indeed, when you provide this synthetic peptide in solution, it does bind to the split fluorescent protein, and after maturation time, it activates the split fluorescent protein as fluorescent reporter. And the interest in here is you can actually control the level of activation by simply controlling the amount of peptides that you put in solution. So you can titrate how many molecules of GFP you want activated by simply controlling the amount of peptides or synthetic peptide you put in solution. So the way we use this split GFP fusion is basically by taking the big fragment right here and fusing it to protein of interest, transmembrane protein or internal protein, and express them in cells, or as I will show you, express them in transgenic animal. And the principle for single molecule is very similar to classical imaging techniques such as PALM, where this time, rather than using the laser, we rely on the stochastic binding of the synthetic peptides, as well as the concentration and time-dependent complementation kinetic to control the amount of molecules that we're going to be activating at the cell surface or in cells. Then we can further localize these individual molecules and track them as they diffuse to extract their diffusive behavior, their position, and information about how they behave in cells and animals. So here's an example of an ensemble imaging in cells. In this particular case, we have split GFP fusion fused to a piece of a CD4 receptor, which is transmembrane protein. To different cells are expressing the split GFP fusion. The negative cells here are not expressing. You can verify the expression level of the split GFP by an anti-GFP staining. And when you provide the small peptides in solution at high concentration, you activate all the fusion protein on the surface, leading to the appearance of fluorescent signal, which co-localize very nicely where the protein is. Notice here in these confocal cross-sections that the activation here is purely at the cell membrane. So the peptides themselves do not activate internal split GFP fusion if they're not directly injected in cells. So this is a bulk assay where you use high concentration of peptide, but as I just mentioned, you can actually control the level of activation by actually lowering your peptide concentration. So here's the same cells with a piece of a cell here, imaged by bright field, and in this particular case, we use total internal reflection for imaging. This is the part of the cell area, fluorescence image of part of the cell area before complementation, before adding the peptides, and within 5 minutes of adding the peptide in solution, as the peptide diffuse, binds, and the fluorescent protein mature, you can see individual spots corresponding to individual fluorescent protein, GFP, CD4 showing up at the surface, and over time the amount of proteins increases as the maturation takes place. You can, again, using Gaussian functions, go from your raw image where you have the individual point-spread function corresponding to the image of the individual molecules. You can localize them by Gaussian fitting and again do tracking and diffusion analysis. In this particular case, we have accuracy in imaging, in localization precision of about 25 nanometer. On a side note, so this is done with a split green fluorescent protein version. On the side note, we are developing alternative FP variants such as CFP variant and YFP variant. We have had big success in cells for the CFP variant, but the split YFP variants are extremely good for single molecule. And in fact, you can see from this video that complementation takes place at the surface of these cells, and you can indeed easily detect individual fusion protein diffusing at the surface. This is again done by total internal reflections. So using these variants, we think we can actually achieve a multicolor individual molecule through the same complementation activated light microscopy technique. The interest in here is that these different color variants all use the same complementary peptide. So with a single complementary peptide, in principle, you can achieve at least 3 colors, and we're working on red variant on our side. I should mention that all these split FP are actually compatible with photoactivable FP if you want to combine CALM and PALM imaging for multicolor detection. So in addition to cells, we use this split fusion for single molecule imaging in animals. In this particular case, we generally do knock-in of the split fusion to receptors or protein of interest, and we express the fusion through endogenous promoter so that the expression is achieved in particular tissues, and then we generally come up with our synthetic peptide, inject them in the circulatory system of nematodes, C. elegans, and as the peptide diffuses through the nematode, it will reach the tissue and light up the fusion at the specific tissue where the fusion is being expressed. So here's an example of fusion to a CD4 receptor. Again, this is a split GFP fusion. This is a bright field image of the worm, and this is the corresponding confocal image in the GFP channels. You can see in this particular case the CD4 fusion is expressed through a MYO3 promoter, so it's expressed in muscle cells. You can see a clear lighting up of the muscles cells upon binding of the peptide. Here's another example which I will develop a little bit later on in the presentation, where the fusion is done through calcium channel. which are expressed on muscle cells again, but also at neuromuscular junctions in C. elegans. Here's an example of a muscle cell where the calcium has been activating after diffusion of the peptides through the worm, again, right at the surface. An interesting comparison is to compare the same calcium channels now through an EGFP, a complete FP fusion. You can see the difference in imaging here. In the case of the SpeedGFP fusion, we light up only the surface channels, while with the EGFP fusion, we also get some signal from the channels coming at the surface, intracellular channels trafficking all the way to the surface. And you can see the difference in terms of the image crispness. In this particular case, here we truly only image the membrane versions of the channels and not at all without interference of the fusion coming from inside the cells, coming at the surface. So these are bulk cases, again, where we have a high concentration of peptides, but something we wanted to do before moving on to single molecule was to actually show that indeed the activation is specifically due to the binding of the peptide. In this particular case, we take advantage of the fact that the peptide is synthetic, such that it can be modified with additional fluorophore. And when you do so, basically complementation will induce activation of the FP, and as well as positioning of a fluorophore in very close proximity to the fluorescent protein chromophore. And in this case, you can actually use the FP as a FRET donor to the chromophore on surface. So in this particular example, we inject peptides modified with the Alexa 647 fluorophore, and we expect activation at the muscle cells to be visible in the GFP channel, but also in the red channels through FRET. So this is the wide field view of the worm, and when you image both the green and red channel by confocal imaging, you can see indeed that the muscles light up, so that means the peptide has been bound, but you can also see that the peptide diffuses through the worm and has bound to different sort of tissues. So there's a lot of non-specific binding of the peptide, which we never see when the peptide is not labeled, but which is clearly visible here upon labeling of the peptide. And so to get rid of these backgrounds, you can actually use these FRETs that I mentioned, where you now excite only the GFP, but you look in the FRET, you look in the Alexa 647 channels, and you actually, as you can see, and I'll go back to the next slide, you can actually see an effective cleaning out of everything that is non-specifically labeled, and the actual muscles light up very nicely in the Alexa 647 near-infrared channel. And an overlay of the GFP and the red channel, usually gives a nice colocalization. So this, for us, is interesting because it confirms that you have irreversible binding of the peptides to the SpeedGFP fusion in animals, and that we can actually get rid of some of the autofluorescence or some of the fluorescence coming from non-specific binding of the peptide in case you use labeled peptide or fluorescently labeled peptide using this FRET detection, basically single per FRET, from the GFP to the dye that is now attached in close proximity. So let's go now to the actual single molecule in animals. In this particular case, we use the same approach where rather than injecting high concentration of peptide, we actually lower the concentration significantly so that we can achieve individual molecule detection. In this particular case, we use a technique called HILO, or highly inclined and laminated optical microscopy imaging, which is a light sheet technique that allow us to, in fact, image muscle cells right at the interface between the muscle cells and the basal lamina within the worm. And as you can see now from this video, when you lower the concentration of peptides to sufficiently low amounts, you can actually detect individual molecules. So the little white spot that you see on the left-hand side are basically individual channels at the surface of the muscle cells lighting up, and they can be tracked. On the right-hand side, you can see the tracking path. The image is a bit lower resolution, but you'll see on the next slides corresponding to tracks of these particular channels at the muscle cell. So we can actually indeed now do tracking. So here's a representative example of different tracks at the surface of the muscle cells. These are example of the signal-to-noise ratio that we have in live animal at the surface of the muscle. Usually we use a probability distribution of the square displacement analysis to identify subpopulation of different protein diffusing. Here are some of the tracks. And the actual localization precision, I forgot to mention that, is about 30 nanometer in the live worm. Our statistics usually are performed on a really large amount of trajectory on the tenth of thousands and in multiple worms, and we can in fact deduce different kind of diffusion mobility in this particular model receptor, which is again this CD4 fusion to speed GFP. We could define basically three types of subdiffusive behavior, 10% Brownians, and the rest of them being slow diffusing in confined domain. So these were model system. What we were more interested in was to apply this to actually disease models, and that's why we focus on this calcium channel, looking at the diffusion at the muscle surface, as well as the neuromuscular junctions of C. elegans model for muscular dystrophy. In this particular case, we take our speed GFP and we fuse it to a piece of the calcium channel called the alpha-2 delta subunit. And similarly, so this is a knock-in fusion, and we replace the endogenous calcium channels with this fusion in worms, and we basically have two types of imaging conditions where we immobilize the worm on beads and where we image the worm between contraction. That's what we call a resting state, and you will see on the right-hand side of this movie a secondary way of imaging the worm, which is through the use of an anesthetic where the muscles is induced to contract. And as you can see again from the left-hand side, the little bright spot that you're seeing corresponds to individual calcium channels being activated at the surface under what we call the resting state. If the movie plays again, you'll notice that the worm is moving a little, so we actually do imaging in between contractions. And then in the case of the drug treatments on the right-hand side, we have the worm being anesthetized, so there's little movement. So you can see here the arrow pointing at some movement, so we usually track in between these contractions. So from these movies, we can now extract diffusion coefficients and information about the trajectory of these different calcium channels. Here I should mention, before I move to the next slides, the fact that we have typical single molecule features such as blinking, which basically confirm that indeed we're looking at individual channels in the live worm. So here are a few examples of trajectories that we obtain. And we can build statistics to analyze the diffusion coefficient of these different channels. In general, we find two population of channel. Slow diffusing channels are versus fast diffusing channel. The slow diffusing channels tend to be confined into 80 nanometers nanodomain, so you can see a clear confinement effect when you do the tracking. And the rest of the populations, which is about 10% of the channels, tend to diffuse freely over short distance around 230 nanometers. So that's and I'll go back a little bit more into details in the next slides, in a few slides, about how we can now use this diffusion coefficient to compare the behavior of the channels in a normal worm and a dystrophic muscle. But before I move to that, I want to show you that you can also use column imaging as well as dual color to localize more precisely where your molecule is diffusing in a live animal. In this particular case, the calcium channels that we've tagged with this PGFP are expressed in the muscles, but they're also expressed at the synapse, at the neuromuscular synapse in the worm. So the worms have these long nerve cords along the body, where muscle cells actually send projections and make synapse to this nerve cord. And our calcium channels is expressed at the muscle surface here, but is also expressed to some extent at the neuromuscular junctions. And to identify the channels that are at muscular junction, we use dual color imaging through a 2CAM system with 2 EMCCDs. In this particular case, the synapses themselves are labeled through an acetylcholine receptor fused to TAG-RFP. And you can see that these TAG-RFP lineups in a line corresponding to these nerve cords and the synapse themselves are labeled in these red spots. And at the same time as we image the synapse, we can also image in secondary channels the GFP fusion activated by complementation by CALM, and you can overlay the trajectories of the channels with the position of the synapses to identify which channels is actually diffusing at the synapse. And you can do this for excitatory synapse, inhibitory synapse, and so on. So in this case, again, this is now halo imaging with dual color, 2 lasers, and we split the signal through an AND or 2-cam system onto EMC-CD camera, and this is imaged simultaneously. So to go back on a little bit more details on the diffusion properties of these channels in normal and dystrophic muscles or contracted muscles, you can see that, as I just mentioned before, these channels tend to be confined. 80% of the channels tend to be confined in 18 nanometers nanodomain. The fast channels themselves diffuse much faster. And then when you compare now this diffusion between resting muscles and contracted muscles, we actually observe a very dramatic decrease in the diffusion properties of the slow population, where the channel seems to be pinned down or immobilized, if you want, by the muscle contraction. And we interpret this, if you want, as a feedback mechanism that is linked to localizing the flux of calcium as the muscle contracts in order to try to potentiate the muscle contraction by enhancing interaction with other kind of calcium channels, other kind of calcium channels that are calcium dependent and that are localized just below the surface of the muscle cells. We also observe a reduced fluidity during contraction through the decrease in diffusion coefficients of these fast channels that you can observe here, which we believe is related to the increased stiffness of the muscle membrane during contraction. And now, to make a long story short, what we observe when we remove or we analyze the same channels in dystrophic worm, where the key protein called dystrophin, which is basically an organization of protein and protein complex just below the membrane that link the cytoskeleton of the muscle cells to the extracellular matrix. When this is being removed, we see completely different behavior of these calcium channels, which rather than becoming slow during contractions, keep diffusing into the nanodomain, indicating that there is probably lack of feedback regulation on the calcium properties by the absence of dystrophin. And then to finish, I'd like just to show you that you can, as Stephen presented earlier, you can actually use all the tricks in the book from single molecule to actually quantify the number of protein per domains or by single molecule counting. So here this is using high concentration of peptides to activate as much as possible of calcium channels at the surface of muscles, and basically going down and letting the molecule bleach and counting the number of bleaching steps corresponding to one particular type of nanodomain. So we sit, if you want, on one type of nanodomain right here and let it bleach up to the point where we reach individual molecule and then count the number of bleaching steps, which allow us to actually evaluate that we have at least 10 calcium channels per nanodomain for the slow diffusing one. We can also use this single molecule localization to do spatial distribution analysis or near neighbors distance analysis, trying to evaluate how are the calcium channels at the muscles of live animals or live C. elegans are being distributed. In this particular case, we found that the distribution of the nanodomains themselves is not random, but is actually more dispersed than random, suggesting that there is a very well organized, very good organization of the nanodomain at the surface of the muscles. And so I like to finish basically by again summarizing the fact that you can use this split GFP and CALM macroscopy to target and image individual molecule in cells, as well as in animal. You can achieve multicolor imaging through the developments of split GFP or split FP variants, but also through FRET processes, or by actually combining with traditional imaging of other FPs. And ideally and potentially this particular technique applied to live animal really allows now biologists to explore the basic principles of homeostatic control as well as the molecular basis of some disease at the nanoscale by allowing imaging in live animal. And with this, I will take questions. This is actually the group in my lab which are working on this particular project, and this is a very nice collaboration we have with Jean-Louis Bressreau and Hong Zhang at the University Claude Bernard in Lyon. These are the funding agencies, and I'm happy to take questions at the end of the webinar.
Thank you very much, Dr. Pinel, for your presentation. It is my pleasure to introduce and welcome today's third and final speaker, Dr. Orla Hanrahan. Dr. Hanrahan is a life science application specialist at Andor Technology. Her role brings her in touch with all the latest developments and innovations in both camera technology and microscopy applications. Previous to this, she has managed a microscopy facility in the biochemistry department at Trinity College Dublin, where she worked with a variety of microscopy setups. Her interest in microscopy stemmed from her PhD in biochemistry, during which she gained experience with numerous microscopy techniques used in her graduate research. Without further hesitation, I would like to hand over to Dr. Hanrahan to begin her presentation.
Thank you, Lynn. I am the final presenter today, and I'm just going to go through some of Andor's solutions for multi-wavelength single-molecule imaging. So both Stephen and Fabian actually mentioned a few of the products that Andor have available for multicolor and single-molecule imaging. And I suppose the majority of techniques now, in terms of microscopy techniques, involve more than just one fluorescent probe. So also, if you're doing live multicolor imaging, you want to be able to, I suppose, minimize phototoxicity to your actual sample. So if you're looking at live cells, for example, and you want to image over long periods of time, and you also want to image multiple colors, you want to be able to use the minimum amount of laser intensity, for example, and also keep your exposure time short that you can get very fast imaging. So using, for example, EMCCG technology with image splitters and two camera adapters will allow you to do this. So we actually have EMCCG and SCMOS cameras available for this for this type of application. And if I just move on then to the next slide, and I'll tell you why we would recommend these particular detectors for multi-wavelength single-molecule imaging. So first of all, it's the field of view. So, for example, this is our latest EMCCG camera, and it's got the largest field of view in terms of the EMCCG technology. So you've got 1024 by 1024 in terms of the sensor format. So that size in itself would allow you to look at three fluorescent probes simultaneously. So if you were to use, for example, the OptiSplit 3, which Stephen mentioned, you could actually attach the OptiSplit 3 to this and still get large field of view images with three different wavelengths. Also, this will allow you to achieve very fast imaging. So if you're doing single molecule imaging with multiple wavelengths, and it's a live application, the faster you can go, the better it is for you. Also, if you're doing super resolution techniques such as STORM, so multi-wavelength STORM, for example, you need to generally acquire thousands of images before you can get one single super resolution image. So the faster you can acquire these individual images, the better it is. Now, the second detector which can be used is the SCMOS detector. And the SCMOS detector is a very large field of view. And what the SCMOS detector can do is it simultaneously gives you very low read noise, very fast frame rates, a very wide dynamic range. And the field of view vary from 4.2 megapixel and 5.5 megapixels. So you've got two different fields of view depending on the SCMOS camera you use. Now, SCMOS cameras generally wouldn't be used for single molecule detection because they're not as sensitive as EMCCG cameras. However, if you're using very bright fluorescent probes, and you generally don't need EM gain if you're using an EMCCG camera, you can easily also use an SCMOS. And the beauty, again, of SCMOS is the large field of view. So you do have the ability of looking at multiple fluorophores simultaneously on one single detector. So that's the detectors. So obviously, if you're going to use a camera, you need to be able to split your light onto the camera, or use some kind of a device which will allow you to split your light onto two different cameras. So in terms of what you could use, the first thing, so both Fabian and Stephen mentioned TwoCam in their presentations, and TwoCam is essentially a two-camera adapter. So you would basically hook your TwoCam up to your microscope, so it can be attached via the C-mount to the microscope, and then you can attach two cameras, one which will collect the transmitted light, or the light that's the far red light, or the longer wavelength light, and then the other camera which will detect the shorter wavelength light. TwoCam can also be used as a switch, so if you did have two different cameras, you could just have a mirror, and you could have one the light going to one detector and then to a different detector for different applications. So you could easily have, for example, an SCMOS on one side and an EMCCDL on the other side. But for multicolor imaging on two of the same detectors, so in this image here, you have two EMCCD cameras, you'd be able to basically get the full field of view from both cameras. Now, you did also see in Stevens that he had an OptiSplit 3 in front of each of these cameras, so you could easily use an OptiSplit 3 in front of these, and then you'll get three wavelengths on each camera. So it is very flexible. There's no device drivers required, so it's just a piece of hardware that you can attach to your microscope, and you can then use whatever software you normally use for your imaging. This is really just to show you the TwoCam. It's very, very easy to use. So the front of the TwoCam, we just recently made this very user-friendly. So generally you would have one camera at the end of your TwoCam and the other camera at the back, so your two light paths. And generally when you're setting it up and aligning your images from each camera, you just need to use these orange buttons on the front of the TwoCam. So you're either going to be turning the horizontal or the vertical, and this will change the position of the image on the second camera so that you can align it on a per pixel basis with the first camera, and then you can overlay your images in them after you've acquired them. The filter cassette then is in the top of this slide, and the filter cassette just holds your filter set, your emission filter set. And this, as you can see, can be just taken out, and you can use it for just regular transmitted light, or you can change the filters if you want to change your wavelength. So again, I won't go through the key specs because we're running a little bit short of time, but it suits the large field of view of the new large field of view cameras, so whether it's an SCMOS or an EMCCD camera. And here is just really showing you the light path inside. So the light path inside, you can see here, this is where the light will come from your microscope. So all of your wavelengths of light will be sent through here, through this adapter, through the TwoCam adapter. It'll go through the lens, and then you have your filter cassette here. And in the filter cassette, your longer wavelength of light will travel straight through to camera 1, and then your shorter wavelength of light will be reflected and will go on to camera 2. And then here at the front of the TwoCam is where you would adjust the position of the image on camera 2 to be matched to the image on camera 1. So it's very, very easy. Now the second device, which could be used as well as the TwoCam, so you could use the OptiSplit 2 or the OptiSplit 3 with a TwoCam, or you could just also put the OptiSplit 2 or 3 on the front of your camera. And the beauty of this is that you only need one camera, and then you can split your light on the sensor of the camera. So an OptiSplit 2 will essentially split your light, so you can use two wavelengths. So an OptiSplit 2 would allow you to look at green and red light, for example, and an OptiSplit 3 would allow you to look at three wavelengths of light on one single detector. So it's an inexpensive solution. It's compatible with a large range of cameras, and it's easily attached to your microscope and to the camera itself as well. And again, just inside the OptiSplit, it's a similar design again. So you have your light, which is coming in here on on this part here. So the light from your microscope, which is being emitted from your sample, will travel in here, will go through the lens, it'll bounce off the dichroic, and the longer wavelengths will travel straight through, and the reflected light will then be reflected, and both wavelengths will then combine and will be split on the sensor. And then in the software that you're using, you can just align the images side by side. So it's very, very easy to use. So that's it. I didn't want to spend too much time because the other researchers had a lot to talk about, and it was very interesting. And if you do need any more information, though, please go to andor.com/scientific cameras. You can find out all about EMCCDs, SBMOS, 2CAM, and OptoSplit. So I'd be happy to take any questions at the end. Thank you.
Thank you very much, Dr. Hanrahan, for your presentation. I can see you've been busy sending through questions for our speakers. Our first question is it possible to identify the position of proteins and particles using FRET? If the protein molecules are encapsulated or bound on the surface, a signal difference is expected, and how do you normalize the signal? This is for Dr. Webb.
So what you're seeing in your image is a single spot. Within that spot, you can't say where each molecule is. You just have a spot which corresponds to the fluorescence from your acceptor molecule and your donor molecule. You can only say how far apart they are from the FRET efficiency, essentially. So yes, you can say within the spot how far apart they are, but no, you can't sort of localize it more precisely.
Thank you, Dr. Webb. Now we have a question for Dr. Pineau, and it is the split GFP used in CALM the photoactivatable form of the protein, or are you using the M3 peptide binding and biting to get single molecules?
So actually, no. So this is not a photoactivable form of the protein. It is really the binding of the peptide that leads to the lighting up of the molecule. In fact, we have strong evidence that once bound and once the protein is mature, the actual peptide does not come off. So it seems to be an irreversible binding, and the imaging is possible until the fluorophore itself photobleaches.
Thank you, Dr. Pineau. One last question before we run out of time is for Dr. Hanrahan, and this is, how do you synchronize two cameras for a single molecule imaging?
Okay, so in terms of synchronizing two cameras, you want to ensure, obviously, that you're going to collect your images simultaneously and at the exact time. So you could externally trigger both cameras to start their imaging at exactly the same time, or you could use a master-slave setup, but you might have a small delay between the acquisition. Maybe Fabian or Stephen might be able to tell how they do the synchronization of two cameras in their setup.
Yeah, we use there's an output on one camera that you can use as the synchronization trigger to the other camera. Synchronize the two, and we use LabVIEW to control our Exxon cameras.
Same thing for us, yeah.
Okay, yeah. Okay, so that's a master slave setup that you use. Okay, yeah, that's fine.
So I'm afraid that's all the questions we have time for, and thank you for your participation in submitting some excellent questions. Any questions that we were unable to answer today due to time constraints will be passed on to the speakers, who will then try to contact you later with an answer. I'd like to take this opportunity to thank all of the speakers for their presentations, and to also thank the attendees of today's webinar. This has been a very informative webinar with a lot of thoughtful questions and answers. Don't forget, there will be a recording of this webinar that will also be available online very shortly. Thank you, everyone, and hope you have a good day.